How about if we just get...
Why are Johnny racing?
This is me and my partner Haruto for the Mitosis project. So cell division is for growth, repair, and replacement of dead cells. The cell cycle is the cell from its birth to the death. Cell cycle is divided into 4 stages or 4 parts. 3/4 of the part is interphase, and the fourth or the last bit of the cell cycle is the mitotic phase. There are 3 sections in interphase. G1 is when the cell gathers scientific stuff. G2, synthesis, or S, that's the code for direction. And the last stage, or like the third stage of interphase, is G2. Again, this is where the cell put together the organelles. So, here you can see the blue string is the chromatin, and the yellow is the nuclear envelope, and the red is the plasma membrane. So when the chromatin duplicates, it becomes chromatics, which are the short blue string. So as you can see, Haruto is organizing these two pairs. So there's a copy of each because you want the new cell to have the same materials. Therefore, now we have chromatics. It has to be together. The chromatics, it has to be together because there's a centromere, and then which makes them the chromatics. Now let's move on to prophase. Prophase is the first stage of mitosis. So in prophase, the nuclear envelope breaks down or disappear, and spindle viper starts to form. The white string here are the spindle vipers, as you can see. And then our pink nucleus in prophase will also appear at each end of the cell. Let's go to metaphase, the second phase or stage of mitosis. This is when the chromatics line up in the center of the cell. See, Haruto is lining the chromatics up, and it is also when the spindle viper are close or connected to the centrioles. You can see right here. So that's all for metaphase. Let's move to anaphase now. This is the third stage of mitosis. This is when the chromatics separate from their parents or break down. As you can see, breaking down the chromatics. And also in anaphase, the spindle viper will draw chromatics toward the central. You can see it is dividing into 2 sides right now and preparing to divide the 2 new cells. Let's get to the last part of mitosis. It's telophase. This is when the spindle viper disappear. You can see now spindle vipers disappearing, and the nuclear envelope, the yellow string, will start to reform, like in the beginning. Now we have our 2 nucleus right there, as you can see. Now, individuals' chromosome will disappear and become chromatin. Same for both sides, because you want both to be the same. Chromatin is a combination of DNA and proteins molecules. There we go. Now we have our 2 nucleus. Now, this is when it starts to divide into 2 cells. So as you can see, there are now 2 cells for the cell division. Both cells have about the same size and the same materials. Let's get to cytokinesis. This also relates to the part of when the cells start to divide. So in plant cell of the cytokinesis, the cell wall will develop inside and then closes off the plasma membrane, while the animal cell, which is closes off the plasma membrane. So there you go. That's mitosis. Thank you for